We call it the high fertility cycle. And if you can get the herd into what we call a high fertility cycle, then you minimize the number of cows that have issues with a deep fat loss after calving.
Welcome back, listeners, to the Dairy Science Digest. This is a podcast designed to bring the Journal of Dairy Science straight to the ears of dairy producers. I'm Reagan Blue from the University of Missouri Dairy Team, and each month I feature a fresh new article in press waiting to go to print. And for this August edition of the podcast, Dr. Keith Latham and Richard Pursley joins us from Michigan State University. And we're going to discuss an article that's currently in press titled "The Effects of Early Lactation Body Condition Score Loss in Dairy Cattle on Serum Lipid Profiles on the Oocyte and Cumulus Cell of Transcriptomes." So really, these titles are so complicated, but really what we're talking about is that fresh cow that quickly loses body condition, and how does that mobilization of lipids affect her reproduction? Before we really get going, could you please introduce yourself to the audience?
Hi, I'm Keith Latham, professor in animal science here at Michigan State.
Hi, yeah, I'm Richard Pursley, also a professor of animal science at Michigan State, and we both work in the area of reproduction.
Very good. And I was noticing on the paper that specifically reproductive developmental science program and comparative medicine integrative biology program. So you really delve into the deep end of reproduction. So the research project that you worked on in Michigan looked at cattle that were housed in freestall barns, fed a TMR, and specifically that fresh window or transition window where you looked at the body condition score prior to calving and then again after calving. And you were really trying to find a group of animals that changed that body condition score, and you compared them to animals that did not change. And the producer that you were working with up there in Michigan, they were feeding a additive, a fatty acid additive, to hopefully help prevent them from changing. But you were still able to capture a handful of cattle that did have a body condition score change so that you could capture their granulosa cells from the reproductive tract and analyze what they looked like. Tell us more about the structure of the project and what you were really trying to determine.
Okay, so there's several other data sets, some that we published before here at Michigan State and some at Wisconsin, that evaluated cows that either lost body condition score, maintained body condition score, or gained during the first 3 weeks to a month after calving. And in our case, in our study, our first study here, we evaluated cows that either lost body condition or we called it maintained or gained. We just didn't have very many cows that actually gained body condition during the first month of lactation. So we just pooled those together. But it does look like, from the standpoint of our data set and the other data set at Wisconsin, that cows that lose a significant body condition have lower fertility when you go ahead at the time you want to breed them, which would be most times 2 to 3 months later. So if they lose a lot of body condition in the first month after calving, that translates into lower fertility for those cows later on. If the cows maintain or gain, it appears they have an advantage in fertility over the cows that lose. So we wanted to know why that is. Why would cows have lower fertility if they lose lots of body condition score? And we chose cows that did not have any health events. In fact, in our previous data set, we had cows that had health problems that lost body condition after calving, but we kicked those out of our analysis to make sure that we were comparing only healthy cows that either lost body condition or maintained and gained. So with that in mind, we chose 10 cows from each group is where we started from, actually. And so we had 10 cows that had body condition loss, and we had 10 cows that either maintained or gained body condition. And what we wanted to know was, are the eggs, the oocytes, which most people refer to as the egg, and which gets fertilized by the sperm, right? Are the eggs different in those two groups of cows? And that's one of the things that we can look at to see why there would be fertility differences between those two groups.
Incredible. And so let's kind of rewind just a little bit and emphasize the fact that we're looking at the body condition score change that occurs long before breeding. Really, those first 30 days of milk is when she's going to be losing the condition that you were looking at that made her eligible to be in this body weight loss category that you were analyzing. So you zoomed in on a molecular level. What'd you find in the cumulus cells of those cows that lost excessive condition?
So our focus was on the eggs. We also collected the cumulus cells. So these were the eggs and the cumulus cells that are associated with the egg when they're ovulated. And so we did what's called a transcriptome analysis. That's basically just characterizing all the expressed genes in the egg, or the vast majority of the expressed genes. The idea being that if the egg is affected in some way, that would be reflected in a change of gene expression and potentially caused by a change of gene expression. So having done that, the main things we observed were that there were changes in gene expression that would be indicative of potential negative effects on meiosis. That's the ability of the egg to divide, complete the meiotic division, which generates the gamete. Potential increases in lipid toxicity effects on the egg, and potential effects that would be causing cellular damage, such as excess production of what are called reactive oxygen species, which can be very damaging to proteins and other structures inside of reduced egg quality, which is something we didn't know going in, whether that would be a part of the fertility effect or not. And it appears that it is.
And so you've got all of these serum nephas that are mobilized to give the cow the fuel that she needs in early lactation that could be creating this kind of toxic environment for the oocyte or the egg to be exposed to. I know some producers like to see the transition cow maybe experience one heat event prior to breeding. Is that some way that we can kind of trump this situation, or is the exposure to those nephas in the body of the cow damaging more than just that first oocyte?
Well, I'm not sure it's a direct effect of the nephas in this case. The Keith can talk about that a little bit more, but it clearly the nephas in about 6 or 7 of the 10 cows and the cows that lost significant body condition were much greater, even at the time of AI. So we took a sample of one blood sample just before we inseminated these cows, and the cows that lost significant body score, most of the cows still had high nephas compared to the cows that were in the maintain and gain group. Keith, you want to comment on the potential effects of high nephas on fertility?
Well, high nepha. So this is one of the Goldilocks things with egg biology. The egg needs a certain amount of available lipids in its environment. An insufficiency of lipids is bad, but an excess of lipids can also be bad. So it needs just the right amount. So excess nephas can be deleterious to the oocyte. Other lipids can be deleterious to the oocyte or to other cell types. And we did see some other alterations in some of the other serum lipids as well. There's still a lot that's not understood about how different lipids affect oocyte biology or the biology of any cell. It's still very much an active area of study. So that's why we decided to characterize the serum lipid profile in a little more depth to get a little bit better understanding of what might be going on. But ultimately, looking at the gene expression pattern of the egg will tell us something about whether the egg characteristics are likely to be different as it appears to you. So.
And then all these cows, at least when we do these studies, it's not an ovulation problem either. So it's
Yeah, right. And you guys only selected the ones that responded to the double off sync.
I think the other thing to keep in mind is we looked at a select set of panels of lipids. We only see changes in something if we looked at that something. There could be other somethings
Sure.
we haven't looked at. So I don't think the goal here was to define specific markers, diagnostic markers to look at. The goal was just to get a little bit better idea of what's going on with the serum lipid profile and ask whether that correlates with changes in the gene expression profile. If you go look at figure 6 of the paper, this was an effort to try to connect some of those changes in serum lipids to downstream effects on the gene expression pattern. And so that's the changes in the lipids are reflected there at the top of that figure, and then they're connected through basically, through a calcium signaling pathway to a lot of downstream pathways from there for which we saw changes in gene expression. And then at the bottom of the figure, it summarizes what functions inside the cell might be impacted by those upstream changes.
It does a really nice job also discussing about the inflammatory response and the fatty acid metabolism and the depolarization of the membrane, just a cascade of things that can occur when the cow has that negative energy balance. And so for boots on the ground dairy producers, what would be your recommendation on how could they potentially prevent this cascade from ever beginning?
Yeah, well, we call it the high fertility cycle. And if you can get the herd into what we call a high fertility cycle, then you minimize the number of cows that have issues with a deep fat loss after calving. So what I mean by that is if cows become pregnant earlier in lactation, and there's obviously a point in lactation, I don't think any of us know exactly what that point is, where there's a break-even point for whenever cows can get pregnant too soon, and then that costs you on the other end of things. But in at least in our hands, if cows were pregnant before 130 days in milk, they had a much less of a chance of gaining body condition later in lactation and being too fat at calving. And so the key is to have cows pregnant before 130 days in milk. And that limits the number of cows that get too fat. If cows go, as you know, as cows get too far out in lactation when they become pregnant, those cows are going to have a longer lactation, and they're going to have a longer lactation with less milk production. And when that happens, they're going to gain body condition, and they're going to be too fat at calving. Those are the cows that cause these issues to occur. And it's mostly in older cows, not younger cows. And first lactation cows, we didn't see as big of a difference in some of these studies, but that's why we chose second plus lactation cows for this particular study.
Absolutely.
So that's key. And if you can get cows pregnant before 130 days in milk, they're about somewhere between a 2.7, 2.8 and a 3 body condition score at calving. Those cows are going to lose less body condition or may not lose body condition at all. And they're going to have a greater chance of being more fertile the next time you breed them 60, 70, 80 days later after calving.
I love the title of a high fertility cycle that you were referring to, and it really is all related. And it's to get that call from a dairy producer that is concerned about reproduction, but oftentimes they call too late. The whole herd is well beyond 200 days of milk, and it's hard to get them to turn around. And at that point, you often see them gaining that body condition score, and it starts to spiral.
No doubt, that's a bad situation for herds to get into. And the key would be to find a program that they can use to get as many cows pregnant on first AI as possible. And what we recommend is one of 3 fertility programs: double OVSync, the G6G program, or pre-sync 11 OVSync, which are all timed AI programs. You can achieve this with breeding to an estrus with those programs. You can breed to an estrus after they come back in heat 21 days later, but not during any of those 3 programs. So they all have to be timed AI. There's a good reason for that. We won't get into that today, but they all have very high fertility for first AI, and that's critical. If you start out with those, you have very high fertility to begin with, and you have, for example, between 45 and 55% pregnant after first AI, then it's much easier to get more cows, the rest of the cows pregnant, or at least a good percentage of them before 130 days. Our goal is to at least have 80% pregnant before 130 days in milk.
I love that benchmark. That's definitely something we can all go back to our records and reference to make sure that we're achieving that. Because if you don't, then we're going to start this spiral of negative energy balance, which your research here published in the Journal of Dairy Science this month is showing has gene expression differences that changes the egg of your cow and prevents her from having high fertility. Very good. So based on the outcomes of your study, what are the reasons for the differences between the fertility of these two groups?
Yeah, is it egg quality? Is it egg quality? Is it reproductive tract? Is it some parameter of maternal physiology? We're trying to flesh out the knowledge there a little bit more, and basically, it looks like the oocyte quality is part of the effect. Maybe not all.
It does look like that the oocyte is part of the problem. Do we think it's all of the problem? We don't know the answer to that, but it certainly is part of the problem.
And this was one study. This was potentially the first study. You mentioned looking at subsequent cycles. One can envision doing a similar kind of analysis on oocytes collected in subsequent cycles to see if oocyte quality recovers. You know, is it a transient effect or is it a more long-term effect? I think it's still an active area of study, basic oocyte biology. How long do these negative impacts in the ovary persist in the oocyte population? Do they get corrected, or do they, persist and give long-lasting effects on egg quality? I think that's very much an active area of study still.
That would be a really interesting future project. As you were discussing that, I have to wonder, I know some producers are gaining rate by utilizing embryo transfer. And so I was wondering if you could speak, and I know this wasn't looked at in your project, but it just in this discussion, it's kind of stirred my thought. I wonder, is there something that's occurring in the cow that's making her more fertile or more capable of carrying that embryo that's past the phase? Is that why we're seeing improved fertility, or I guess pregnancy rates with embryo transfer, do you think?
Wow, that's a great question. I think it's possible. Now, we only used the older cows in this study, the second plus lactation cows. So that being said, those cows were the ones that were the problem with regards to fertility following this body condition loss. And when you bypass all that, that's one way to bypass that, is to put an embryo in that cow, a number one grade embryo from obviously another cow that would bypass that and potentially improve fertility. That is a study that has not been done, where they've taken cows with extreme body condition loss versus none, and then put in embryos just to see if you can alter the impact of that, which you would think that the embryos would have much better fertility than the potential for the oocyte that's having these issues. So no doubt that would be an alternative. If you want to improve first service conception rates, that would be an alternative method for sure. That's a great question.
Perhaps by doing that, you would be able to determine if it's an inflammatory response effect or exclusively an oocyte effect. So just to be clear, I read about early embryonic loss in popular press. That's not what we're talking about here, correct? This is going to be long before you ever get the opportunity to believe that she's pregnant. Is that correct?
Well, we don't know the answer to that question, to be quite honest. I mean, we don't know what the studies that are published right now didn't have great embryonic loss information in it. So they had pregnancy data, but they didn't have the follow-up with the embryonic losses. So don't know the answer to that question. And we're certainly interested in those things and are studying pregnancy loss quite a bit these days. But that's a great question we don't know the answer to.
On a conceptual level, reduced egg quality could lead to subsequent embryo demise. Not sure when that would happen, or whether it would be before implantation. It could potentially happen after implantation. There's a whole field developmental origins of disease that would posit that things that happen very early in the oocyte or embryo can have a big impact on even adult phenotype and progeny.
Mm.
Good, real good point.
Getting pretty far afield there, but
It's still a bit of a black box, isn't it?
well, kind of. I mean, some of the work by Tom Fleming looking at effects of mild protein restriction in maternal diet. If you reduce the protein content of mom's diet by half during the pre-implantation period and restore a normal diet, progeny wind up being hypertensive as adults. And it's believed that that very mild change in protein content creates a stress, and then the embryo adapts to that stress, but that adaptation to that stress is kind of reversible.
Yeah.
So then when you change the diet back, now the embryo is not optimized for the new environment that it finds itself in. So that leads to downstream effects potentially on stem cell populations and things like that that can affect the development of the kidney or other organs. So it's a black box, but it's maybe more of a gray box. We have some inclinations of some possible mechanisms, but I don't think anyone has really mapped out specific sequelae of events to show how to show how these things work. But
I applaud both of you guys for your research, gathering up pieces and parts of the puzzle that will help shape the discussion as we move forward and directly impact the reproductive success of our dairy herd. And this has been incredibly informative, and I really want to thank you for your time. And listeners, I applaud you for taking time out of your day today to learn about how your early lactation change in body condition score could impact the oocyte of your open cow that you're trying to breed back before 130 days. So I've really enjoyed our conversation learning about reproduction on this molecular level. This has been the August edition of the Dairy Science Digest, which is a monthly podcast project designed to bring the Journal of Dairy Science straight to your ears. We highlight peer-reviewed research articles in press, sound science that you can base your management decisions around, provided by your University of Missouri dairy team. Be sure to like, subscribe, and share to get future editions straight to your cell phone. This is Reagan Blewell with the Dairy Science Digest, and I hope you have a great day.